Hi there. I'm going to give a quick presentation on an organism called Naegleria fowleri, which is sometimes colloquially referred to as the brain-eating amoeba. So let's get started. So what is Naegleria fowleri? Well, it's a thermophilic, single-celled but eukaryotic free-living amoeboid which can be found all across the globe in freshwater lakes, ponds, and streams, and sometimes even in more man-made environments such as water treatment plants or pipes in homes if not properly cared for and chlorinated. It's not a true amoeba, and this is partially because it is able to take on different body plans and sort of shapes over the course of its life cycle, which we can see demonstrated over here in the diagram. The three forms that Naegleria can assume during its life cycle is a cyst, a trophozooite, or a flagellated form. In a broader sense, it's a member of the phylum Percolozoa. It's the only member of this phylum that we know of that's infectious to humans. It's often referred to in the literature as well as an excavate or a member of the group Excavata, which is, I believe, a now defunct classification of organisms, but it still appears a lot. So to give you sort of a broader context, in this cladogram, we can see this blue arrow over here is pointing to N. fowleri, and next to it is Naegleria gruberi and Naegleria minor, which are other species in the genus. And you can also see a little further down, there's the Valcampia, which is sort of the cousins of these amoeboid organisms. And obviously there's a ton of other species and organisms related to them which have been described. But Naegleria is the sole among them, which is infectious to humans. It was discovered by this gentleman over here, Malcolm Fowler. It's the namesake of Naegleria fowleri. Fowler was a histopathologist, actually the director of histopathology at Adelaide Children's Hospital in Australia, and he first described this infectious microorganism, or this particular amoeboid, when diagnosing a child in Adelaide with the infection. Naegleria, the genus, though, and organisms like it have been described since 1902, early around the turn of the century, when this amoeba was noted and differentiated from others as being able to take on a flagellated form depending on its environment. Other members which had been described prior to Naegleria fowleri were again N. gruberi, mentioned above in the cladogram, and N. leviensis. Prior to the advent of genetic sequencing technology to differentiate between species, it was really common to morphologically distinguish them, and so a lot of what we know morphologically about N. fowleri and its relatives came from N. gruberi, both in terms of examining it under a microscope and in terms of genetic evidence. So let's talk a little bit more about its life cycle. As we mentioned before, it can assume three shapes during the course of its life cycle depending on its needs. If this organism were to find itself sort of in a resource-poor environment, it's able to encyst, which we can see on the left over here. The cyst stage, it actually undergoes some pretty radical cellular remodeling to achieve this. Its cell wall and envelope thickens, as does its cytoplasm. It sort of dehydrates itself and condenses itself down into a smaller form and lowers its metabolic rate. If this cyst were to, for instance, be found in a lake that dries up in the dry season, the cyst might form when the water is no longer available and it's sort of on the shore of the lake in the dust. But when the wet season returns again, rain might wash the cyst from the dust back into the body of water, at which point it would excyst and take on the trophozooite stage, which we're seeing in the middle here. The trophozooite stage is when it's capable of feeding and growing and reproducing. It's also when it's infectious. So this is commonly seen. It can move around a little bit, but not nearly as much as the flagellated stage, which is the final here that we see on the right. The flagellated stage, as you can see, isn't quite as blobby. It's a little bit more hydrodynamic looking, and it has two little wisps coming off sort of the end there, which are its twin flagella, which it uses to move around its environment and seek food. So that's the motile stage of life. And from a flagellated stage, it can turn back into a trophozooite or a cyst as needed. So how exactly does this get into our body to cause disease? Well, as you can see in the diagram provided by the CDC over here on the right, you can imagine a cyst either, as I described, being on the shore of a lake or in dust or in water, excysting and turning into a trophozooite or a flagellated form, depending on what it needs. And at that point, if you were to hop into that lake, pond, or stream and go swimming, and some water were to get shot up your nose, maybe you took a dive, maybe you're just, not swimming as well that day. If it gets into your nasal cavity, this is actually how it's going to enter your brain. This is not the only way that this happens, though. In some cultures in other parts of the world, ritual ablution is a common practice, such as in Islam, whereby you wash out your nasal cavity by sort of aspirating water into your nasal cavity up there. This is similar to what you might do with a neti pot, which again, as we've discussed, this organism can be found in poorly treated water, and so it's important to make sure you are using clean water if you're ever going to be washing your sinuses and your nose out with something that could even potentially be infected or contain these organisms. There's also been some more anecdotal cases of the cyst form in dust being aspirated into the nose, which is scary because that's much, much more difficult to prevent, and you really wouldn't know about it. However, this is even less common than the normal route of entry, and again, mostly anecdotal. So once it's in your nasal cavity, how does it actually enter the brain and cause so much damage? Why is it called the brain-eating amoeba? Well, once it gets in there, it embeds itself into the nasal epithelium, which you can see on the bottom of this diagram, or the pink rectangular cells. They'll sort of get into the mucus and evade the immune system. And from there, it can actually use this network of neurons, which outpockets from the brain into the olfactory bulb and eventually all the way down to the olfactory epithelium. This is how our sense of smell works. And you can see sort of the axons extending out through what the middle part of this diagram is showing, called the cribriform plate. So you can imagine a Naegleria fowleri in the nasal epithelium being able to physically move through these tiny little pores in the cribriform plate in the middle of this diagram, the bone, and sort of follow the axons up to the glomeruli, where they then synapse on the neurons of the olfactory bulb. And then these axons, again, will connect directly to the central nervous system. And so they're able to sort of wheedle their way into the olfactory bulb through the axons and openings in the cribriform plate in a way that you couldn't in other parts of the body. It does this in a couple of ways, which we'll discuss a little bit more in the next slide. But essentially, this is how it gains access to the brain to wreak so much havoc. When it does this, it causes something called primary amoebic meningoencephalitis. This is how the disease is characterized. Primary amoebic just means the primary cause of the meningoencephalitis is from the amoeba. And of course, meningoencephalitis refers to an inflammation of the meninges, or the layers of the brain, and encephalitis is a generalized swelling of the brain. So when it's actually getting into the brain, it does this in two ways. There's a contact-dependent and a contact-independent mechanism by which it can enter. The contact-dependent really means it needs to physically interface with the neuronal cells, usually using pseudopods and moving through cells, and also sometimes destroying them mechanically with the use of these pseudopods, which for an amoeba are sort of like, well, pseudopod in Latin means false foot, and so it's able to sort of move aside or potentially destroy cell membranes. But another key part of how it enters the brain and gets through the olfactory bulb is using biomolecules, which it will secrete, which both can damage cells and cell membranes in our bodies and also stimulate inflammation. For instance, some of the biomolecules it secretes will encourage inflammation and allow leaky capillaries, essentially making it easier for cells to exit the bloodstream and enter the brain and cross the blood-brain barrier. This is true of both leukocytes, which will cause inflammation, and N. fowleri itself. And so it leads to a generalized inflammation of the brain, which causes swelling and a lot of damage. So how exactly do you see this manifest? Well, as soon as one day to a couple of weeks after being infected, you will start seeing early signs of PAM, primary amoebic meningoencephalitis. However, these are kind of difficult to distinguish as being caused by an amoeba, both because it's so rare and because they're virtually indistinguishable from other symptoms, such as a fever, a headache, nausea, stiff neck. These could all be written off as just a cold or a flu. Unfortunately, however, sometimes as soon as an hour to about a week or two after these symptoms begin, your neurologic symptoms begin to progress pretty rapidly to seizures, altered mental status, confusions, hallucinations, as the amoeba begins to take over and cause damage all over your brain and central nervous system, both in the form of inflammation and tissue destruction. Once these symptoms have begun, there's really not much that can be done, which we'll discuss a little bit more in a moment. Nearly all cases which have reached this stage, in fact, nearly all cases in general, result in coma and eventually death through brain dysfunction, organ failure, and the inability for you to regulate your body's needs. So this is a graphical representation of kind of what we're looking at here. Once N. fowleri enters the nasal cavity and enters the brain, it uses contact-dependent and independent methods to stimulate inflammation and infection. You can see some of the white blood cells, leukocytes, and cytokines that it will recruit in the process that sort of contribute to the infection that will cause the swelling and inflammation of the brain. And down at the bottom here, we'll talk about this a little bit more in a moment. We have the two current treatments, generally speaking, that we look at, amphotericin B and miltefosine. Again, these are not proven to work, in very, very few cases result in survival of this infection once it's progressed. So how would you diagnose this? As we mentioned, it's very difficult to clinically distinguish this from other diseases, both, for instance, a bacterial meningitis, or in the early stages, even something like a cold or flu. So it's very difficult to do and identify early on. And oftentimes these cases are done post-mortem, only after you've died, and we're trying to figure out what the actual infectious causative agent was. If you were to correctly identify it or test early enough, the way you do it is by obtaining a sample of cerebrospinal fluid by a lumbar puncture and other means, and visualizing the fluid for any potential cells. You can see here this diagram, the arrows pointing to the trophozoite stage of N. fowleri in CSF. So if you were to go through that and you correctly identify it, you get your CSF and you find the organism in time before you've progressed to a coma and death, how would you treat it? Well, as we alluded to earlier, we have two main methods, amphotericin B and miltefosine. Amphotericin B is an antifungal that's used pretty widely. How exactly it can prevent or kill N. fowleri isn't entirely clear, but in other cases treating fungal diseases, it likely interferes with metabolism and the formation of the cell wall, which has some mechanism, it's thought or postulated to be able to affect Naegleria as well. Miltefosine is another drug which is usually used to treat leishmaniasis, and again, this probably acts by disrupting membrane integrity of these organisms such as N. fowleri by inhibiting some metabolic pathway. This drug is not FDA approved, and so if you suspect or have a diagnosed case of PAM, then you would need to request this drug specially and hopefully get it in time for it to work. So where does this actually happen? Where are you at risk? Where do we find them? You can see here sort of a heat map. As mentioned, because this organism is thermophilic, it's concentrated sort of near the equator and in warm climates. You can see some cases, however, that are spreading closer to the poles in places that are not quite as warm. For instance, up in the top left there, you can see an arrow pointing to Adopt in Minnesota. This is a case that was documented maybe over a decade now of a little girl who unfortunately died from this disease that far north. She was swimming in a lake in Minnesota. And it was surprising because, well, as you can see, there's many well-documented cases in the southern United States. Seeing it so far north was uncommon. This is something that epidemiologists and parasitologists and biologists around the world are aware of. The range of thermophilic organisms such as N. fowleri will expand as global warming expands, and so it's not unreasonable to imagine that we will begin to see more and more cases in places we wouldn't otherwise expect to see them in the coming years. And of course, part of this seeing more cases or high reporting of cases is not just that their range is spreading because of global warming, but also because we're getting better at identifying it, we're getting better at testing for it when we do have a potential case come in, and we're getting better at looking for the organism in the environment. So whether or not the increased incidence is due to just higher rates of testing or actually a broader distribution is difficult to say. So given that this drug has a 95 or greater % mortality rate, it's currently not treatable, and it seems to be that its range is spreading potentially to a warm pond, lake, or river near you. How can you prevent it? Obviously, we don't want to slip into a coma and die, and so the best way that we can prevent this is actually just to prevent getting water in our nose. It's important to note, however, that N. fowleri infection is incredibly rare. There's been less than 500 cases in the last 50 years or so, and while the vast majority of them have led to death, it's just not that likely to happen. But as you can see in this diagram here, something like a nose clip is an effective way to prevent water from entering your nasal cavity if you are swimming in, for instance, the southern United States in a freshwater lake or pond. It's a very small thing you can do to sort of prevent the risk of getting this infection, which again is incredibly rare and not something you should generally be concerned about, but be cognizant of. So thank you. That's my presentation on N. fowleri. Again, there's lots more to study and lots more to know on this organism. Specifically, how it is infectious and unique as an organism is very interesting. And of course, importantly, its range, spread, how it's going to be impacted by global warming, and how we can better address these infections and hopefully combat them before they spread further. So thank you very much for listening. I hope you enjoyed.